PhD, and then he wanted to join my lab, and I asked, "I hope you like copepods." He says, "I love copepods, but not your boring copepods because they're too easy to collect. They're too easy to keep in lab, and their genomes are too small. I'm going to go for 25 miles offshore to go collect copepods. They're much harder to find, much harder to maintain in lab, and whose genomes are 3 times bigger than a human being." So he made it really difficult, but he spent the first year and a half. That's something he's not going to say, have time to say, but he spent first year and a half improving the methods for maintaining these copepods in the lab to the point where he was able to do a lot of experiments in them for the first time in the system, doing a lot of experimental work, genetic work in the species. So I'm very excited about the progress, but I wanted to emphasize how much Kris has been a force in the, I think, in the IB community as a model for just independence and excitement for marine sciences. And it's and he's still going to stick around. He's going to be at Hatfield for postdoc, so he's not going anywhere for a while. But I'm excited to have you all hear his research. Great.
Well, thank you all so much for coming today. Thank you, Felipe, for such a kind introduction. And I just want to say it's really amazing to see so many familiar faces from all different walks of my life here in the audience today. And names popping up over Zoom. So thank you for taking the time to be here today. And I hope you enjoy my talk. So the title of my presentation is Evolution in Upwelling, the Adaptive Capacity of a Key North Pacific Plankton Species to Climate Change. I'd like to begin by sharing the driving question behind my graduate research over the last 5 years. How are plankton populations in the coastal Pacific Northwest being impacted by climate change? And I chose to seek answers to this question through 3 different projects. In projects 1 and 2, we focused on plankton populations on the coast of Oregon. In project 1, I focused on changes in the physiology, and in project 2, I focused on the changes in gene expression in response to environmental change over a single summer. And then in project 3, we take a broader view and look at plankton populations throughout the coastal Pacific Northwest to see how environmental heterogeneity drives genetic differences between populations of plankton throughout this range. But before I begin, it's important that we all have a firm understanding on what I'm talking about when I say the word plankton. Plankton, the root meaning of the word plankton means to wander or drift, and it refers to a type of organism that cannot swim against the current that it lives in. So to put this into perspective, I want to show one of the fastest moving currents in the world. This is the Gulf Current that extends out of the Gulf of Mexico and moves across the Atlantic Ocean, moving from west to east. Plankton caught in this current, I don't know, laser pointer, hold on. Plankton caught in this current cannot swim against the direction that that current is moving. So they are drifting passively as this current moves across the Atlantic. But that doesn't necessarily mean they're poor swimmers. In fact, many plankton can swim up and down in the water column. They just can't swim horizontally. There are 2 general kinds of plankton, phytoplankton and zooplankton. Phytoplankton refer to the photosynthesizing plankton. They have been on our planet for a very long time and still play a crucial role in the production of oxygen. In fact, they produce over half of our planet's oxygen. That means that every other breath you take is thanks to phytoplankton. Ecologically speaking, in marine food webs, they play the same role as plants in terrestrial food webs as primary producers. That means they are converting sunlight into energy, into calories, that is then transferred up the food chain all the way to apex predators. Each level on these food webs is known as a trophic level. Organisms that inhabit higher trophic levels are typically predators, and in the ocean, this includes many of our fishery species. And then these organisms that inhabit lower trophic levels are also very important because they are transferring this energy from primary producers to these higher trophic levels. And in the ocean, the role of the grazer is typically taken by the zooplankton. In general, these are the non-photosynthetic plankton, and similar to phytoplankton, they have been on our planet for a very long time. However, unlike phytoplankton, which are typically very small, they can come in a huge range of sizes. So from microscopic, like these dinoflagellates I'm showing you here on the left, all the way to absolutely massive, like this giant phantom jellyfish that lives in the deep sea off of California, which can grow up to 33 feet in length. That's over half the size of a city bus. So when I'm talking about plankton, I'm not referring to a single lineage of organisms of a specific size or function. I'm talking about a type of organism that has evolved to live and die by the current in which it resides. Life on our planet would be fundamentally different without plankton in it. They play major roles in global biogeochemistry, in the ecology of all aquatic ecosystems, and are some of the most abundant organisms on the planet. Today, we're going to be focusing on a specific group of zooplankton known as the copepods that are widely believed to be the most abundant animals on the planet. With over 12,000 species, this immense diversity comes from the fact that they occur literally anywhere there is water on our planet, even deep underground in geothermal wells or high above ground in tiny pools of water that can form in the canopies of trees. Believe it or not, we've even found copepods in our drinking water, but this is a story for another time. Today, we're going to focus on a specific species of copepod in the ocean known as Calanus marshalli. This is a highly abundant copepod on the Oregon coast, which is connected to a larger current system known as the Northern California Current. This is a highly productive current system that supports an abundance of biologically rich and diverse marine ecosystems from the rocky shore to the deep ocean. Collectively, these ecosystems support over $500 million in revenue to coastal communities through industries such as fishing and aquaculture. And what facilitates this biological abundance is an oceanographic process known as upwelling, in which seasonal winds drive coastal waters offshore to be replaced by cold and nutrient-rich waters from the deep. In the Northern California Current, this happens every year between the months of May through August. And this added nutrients essentially fertilizes the water column and leads to huge increases in the abundance of plankton, such as our copepod here. This high abundance makes them excellent sources of food for many of our fishery species like salmon or rockfish. And what's more is that this is an extremely fatty rich copepod that I'm showing you here. These red blotches that I have circled, those are fat. And essentially that makes it look like a juicy cheeseburger to many of their predators. But they're not just producing these fats to look appetizing, they actually utilize them to survive over periods of time when upwelling is not occurring. Essentially, these copepods dive into the deep ocean and go into a state of hibernation during times of the year when upwelling is not occurring. This is known as diapause, and they live off of these fats during this time until upwelling conditions return the following year, and they return onto the coastal shelf. This makes them highly adapted to living in upwelling conditions. So much so that even though they're plankton, they're very efficient at maintaining their place in the phytoplankton-rich areas of the water column above our continental shelf. Eggs and early life stages occur in these surface, warm, phytoplankton-rich surface currents that are drifting offshore as a result of upwelling. But as they get older, they begin to dive deeper and deeper to avoid being eaten by predators. And eventually, once they reach these later stage and adult stages, they dive deep enough that they actually get caught in these upwelling currents and are brought back onto the continental shelf. So as a result, they're able to maintain their place on the continental shelf and have about anywhere from 3 to 6 generations per summer. But while this life history makes them very good at staying on the continental shelf, climate change may be beginning to change that. While this upwelled water is nutrient rich, it is also oxygen poor, as you can see from this plot here on the right. Here we're looking at a cross section of the continental shelf off of Oregon. Oxygen rich water is shown in blue and oxygen poor water is shown in red. And you can see that it's being brought up onto the continental shelf during upwelling. And over time, the collection of this oxygen poor water can result in a phenomenon known as hypoxia. Hypoxia just refers to oxygen poor water that is essentially very stressful or potentially lethal for the majority of marine life within it. And historically, hypoxia really only occurred on the farthest and deepest stretches of our continental shelf. But over the last 20 years, climate change has been intensifying the winds that drive upwelling, leading to a greater amount of this hypoxic water to move into the nearshore and shallow environment where a majority of our coastal marine ecosystems lie. At the same time, while this oxygen poor water is being brought onto shore, there's also an excess of nutrients that are being brought onto shore thanks to climate change. And as a result, phytoplankton populations are essentially growing faster than zooplankton can control them, leading to a high amount of phytoplankton dying and being consumed themselves by bacteria, which also take in quite a lot of oxygen. So essentially, oxygen conditions start low at the beginning of the upwelling season, but they continue to worsen as the upwelling season progresses. This is all happening at the same time as the Northern California. So at the same time as the Northern California current is becoming more hypoxic every year throughout the upwelling season, we're also seeing a sharp increase in the number of marine heat waves, particularly over the last 20 years. Marine heat waves are periods of anomalously warm conditions that can last for days, weeks, or even months that add further stress to our coastal ecosystem. As a result of this hypoxia and warm temperatures, this has led to multiple mass mortality events in our fisheries over the last 20 years, mainly Dungeness crab. In fact, it was fishing communities that were the harbingers of this environmental change as they started pulling up pots filled with dead crab in the early 2000s. And as a result, it's through monitoring these fisheries that we've been able to infer ecosystem health, both during and after these stressful periods. But as I mentioned earlier, many of our fisheries occupy these higher trophic levels. And I wanted to know more about how climate change was impacting these lower trophic levels, the grazers, the zooplankton, which brings me to my work. So as I mentioned earlier, I decided to go about researching how climate change was impacting zooplankton through 3 distinct projects. In project 1, I focused on the physiological tolerance of marshalli to hypoxia and warm temperatures throughout the upwelling season. In project 2, I focused on changes in the gene expression in response to hypoxia and warm temperatures throughout the upwelling season. And then in project 3, we take a broader view, as I mentioned earlier, and look at patterns of genetic variation, potential adaptive divergence in Calanus marshalli throughout its southern species range. We'll start with project 1. So here, I split my questions based on our 2 stressors, heat and hypoxia. For heat, I asked what is the thermal limit of marshalli and how does this limit fluctuate throughout the upwelling season? And then for hypoxia, I asked what is the lower oxygen limit of the species and how do these limits fluctuate throughout the upwelling season? All with the intention of getting a better understanding of what metabolically accessible space looks like in the water column for this species throughout the upwelling season. So in order to answer these questions, I was lucky enough to go out with the Peterson Lab at Hatfield Marine Science Center off the coast of Newport on their monthly plankton surveys from May through August of 2023, where I sampled environmental conditions of the water column using a CTD, or conductivity, temperature, and depth sensor at 5 different oceanographic stations, shown here by these green icons, each 5 kilometers apart from one another. And then I sampled plankton at these farthest 2 stations, 20 and 25 kilometers off the coast of Newport, using these nets, which I then brought back to shore into our lab and acclimated these. So I picked my copepod out of this greater plankton sample and then acclimated these copepods to lab conditions for 24 hours at the average depth of the water column during the upwelling, or the average temperature of the water column during upwelling, and then fed them a mix of different algae. After that 24 hours, we started our experiments split based on heat and hypoxia. I'll start with heat. So here, we took 10 copepods from these food-rich conditions, placed them into these tubes called falcon tubes individually, and then placed all 10 tubes into a water bath at a specific temperature for 1 hour. And then after this hour-long exposure, we placed them back into ambient conditions and monitored mortality 24 hours later. If a copepod was alive, that meant that it could recover from that thermal stress. But if it was dead, that means that that thermal stress was likely lethal for that copepod. We repeated these experiments at 5 different temperatures, each with a different group of 10 copepods every time. So what we did with this data is we were able to feed it into a model that could predict the probability of recovery or mortality 24 hours after exposure at each exposure temperature here on the x-axis. So this is what it looks like for May. So a probability of 1 represents a high probability that that copepod would be able to survive would be able to recover after exposure, whereas a probability of 0 means that there is a very low probability that that copepod would have been able to recover. And essentially what we're measuring here is what's known as the median lethal temperature, or LT50. This is the temperature at which the predicted probability of survival reaches 0.5. Essentially, when it becomes more likely that the copepod will die rather than recover from this thermal exposure. And for May, this temperature was right around 24.6 degrees. Unfortunately, for June, we didn't get the samples to perform this experiment, so we had to omit this month. But in July, our LT50 was 23.9, and August, 24.2. Essentially, that means that the thermal limit of this species is right around 24 degrees, and it doesn't really vary much throughout the upwelling season. Moving on to hypoxia, we utilize what are known as respirometry trials. This essentially includes taking this really cool, fancy glass plate that has 24 wells in it, and in each well is an oxygen sensor. And we could fill these wells with seawater and a single copepod and measure in real-time oxygen consumption, which I'm showing you here on the right. So when we start these trials, we seal the plate to make sure that no oxygen from the atmosphere can get into each well. And essentially, as the copepod respires this oxygen to fuel its basal metabolic rate, oxygen decreases in the well until eventually it gets to a point that oxygen becomes so low that it can no longer sustain that copepod's basal metabolic rate, and that metabolic rate crashes. The point at which it crashes is known as the p-crit value. And that is what we are measuring in order to quantify hypoxic tolerance within and between individuals each month. We performed these respirometry trials at 8, 12, and 15 degrees to see how temperature impacted p-crit throughout the summer. And here's what we found. So here I'm showing you a violin plot of our p-crit values from May through August, all at 8 degrees here. And as you can see, over time, we start with a relatively low p-crit value here in May. But as the season progresses into August, our p-crit value is significantly higher. This means that copepods are becoming more sensitive to hypoxia throughout the upwelling season. So a low p-crit represents a high tolerance to hypoxia, and a high p-crit represents a low tolerance to hypoxia. There was we ran ANOVA, which is essentially a statistical test to determine whether or not this variation that we're seeing could just be driven by random processes, or in fact, whether or not there was a biological component to this difference. And we found a statistically significant difference, meaning that the probability that these differences that we saw, just being based on random variation, are very, very low. At 12 and at 15 degrees, we see very similar patterns here, where p-crit starts low at the beginning of the upwelling season, but ends much higher at the end of the upwelling season. Essentially, this means that Marshallite starts with a robust response to hypoxia early on in the upwelling season, but over time, that response is largely degraded. And so whenever, in terms of their lower oxygen limit, it really depends on when you sample throughout the upwelling season. So as I mentioned earlier, we can take this data to get a better understanding of what metabolically accessible space looks like in the water column for this species. In order to do this, we can use what's known as the metabolic index, which is essentially a ratio of oxygen supply to oxygen demand. Supply represents the environmental oxygen levels, which I mentioned earlier. I took from the water column every month using the CTD, and then oxygen demand is represented by the temperature-specific p-crit values that I just mentioned earlier. And these can give us values that indicate or give us an indication of what is a likely stressful environment for this copepod. So values of 0 to 2 represent areas of very high stress, potentially lethal. Values from 2 to 4 represent areas where the copepods are likely beginning to get stressed or that stress is building. And then values of 4 or above represent areas where there is plenty of oxygen for that copepod to support metabolic demand. So using these values, I can actually create heat maps of the water column, which I'm showing you here on the left. This is off the coast of Newport. The top is the surface. We're going down to a depth of 140 meters. This is the average depth that we find Marshallite at between the surface and 140 meters. And then this black polygon here represents the seafloor. These data are actually from May. And as you can see, the upper area of the water column has plenty oxygen to allow copepods to maintain their metabolic rates, whereas these deeper areas of the water column may begin to get a little bit stressful from marshali. But as upwelling progresses into June, this low oxygen water occupies this deeper area of the water column, leading the entire water column to be overall more stressful. And then this pattern continues into July. And by August, we're looking at a completely different water column in terms of accessibility for this copepod than we did in May. And so this question then becomes, why this change in tolerance? Why do we think we saw what we saw in terms of this variation? And we believe it's because copepods in August are spending a much greater period of time in this stress than they did in May. So essentially, that stress compiles over time, leading to overall, essentially keeping these overall, excuse me, leading to these copepods essentially not being able to mount as robust of a response as they did early on in the upwelling season. And so now we're interested, so with this in mind, we're interested in how specifically does this stress tolerance weaken and by what molecular processes? To answer that, we can look to project 2, where I look at changes in gene expression. So here, again, I split my questions between heat and hypoxia. For heat, I asked how do the patterns of gene expression change for thermal stress, and do these patterns also change throughout the upwelling season? Similarly, for hypoxia, in response to hypoxia, how do patterns of gene expression change, and do these patterns also change throughout the upwelling season? But before I go any further, I want to clarify what I'm talking about when I talk about gene expression, and I'm going to use OSU's mascot, Benny the Beaver, to help me with that. So let's pretend that Benny wants to build a dam, but in order to build that dam, he needs log proteins. He's got thousands of log proteins that he needs to help him build this dam. We're going to focus on one log protein specifically that's produced by the log gene. This log gene activates and transcribes the RNA, which I'm showing you here by these sticks. And then this RNA is translated into our log protein, which, as I mentioned, there are thousands of different kinds of log proteins here that once he has all of them, he can build his dam. But now that Benny has his dam, he doesn't need to produce RNA from this log gene anymore, essentially deactivating that gene. And we call this downregulation. So in this case, the log gene activity is going from high to low, and that's what downregulation refers to, when the activity of a gene goes from high to low. But now let's talk about the case where Benny wants to build a bunch of dams. In that case, the log gene activity is going to increase really quickly, leading to a bunch of RNA, which are then translated into a bunch of log proteins. This is known as upregulation, where log gene activity starts low, but then increases over time. So essentially with my copepods, what I'm asking here is, in response to heat or hypoxia, what genes are downregulated and what genes are upregulated in response to this stress? I used the same populations that I collected in May and August. I want to clarify, I only looked at gene expression in these 2 months specifically, because we saw the largest change in physiology between these 2 months. I acclimated them the exact same way as I mentioned in project 1, and again separated my projects based on my data collection based on heat or hypoxia. So for heat, similarly, we're going to work. Hello? There we go. So for heat, similarly, we started with 10 copepods, but this time we had 2 different treatments, a heat exposure and a control. So we took each individual copepod, placed them into falcon tubes. Some of those, or half of those falcon tubes, went into a warm water bath, which was our heat exposure, and some went into a water bath that was set at the same temperature that they were already at, essentially being our control. And then after an hour of exposure in at each temperature in each bath, we collected RNA. And then for hypoxia, similarly, I had 2 treatments here, a hypoxia exposure treatment and a control. And I utilized my respirometry plates, but in a slightly different way here. So just like in project 1, for my hypoxia exposure treatment, I sealed the well so that oxygen would decline over time as the copepod expires. But for the control, I just left the well unsealed so that oxygen could maintain or could be essentially sustained, kept at a high level throughout the trial. And then over time, as that copepod in the sealed well got closer to peak crit, I sampled both individuals in the control and the hypoxia treatment at the same time, collecting RNA. So here's how we visualize our results from gene expression. We use a specialized scatter plot known as a volcano plot. I have no idea why it's called that. And essentially this gives us a measurement of how significant the differences were in gene expression and whether or not they were upregulated or downregulated between treatments. So on the y-axis, we're looking at statistical significance, which increases as you go up. And then on the x-axis here, we're looking at the log of the fold change, essentially how much a gene is upregulated or downregulated. Upregulated genes are shown on the right, and downregulated genes are shown on the left. Each point here represents a single gene, and we only want to focus on the red points here, because those are the genes that were significantly upregulated or downregulated between treatments. So these are actually our data from the heat shock experiment in May. And as you can see, a large number of genes were significantly upregulated, where only a small number were downregulated. But beyond the number of genes that were differentially expressed, it's also very important to know what the function of these genes are. And so for this, we can use what's known as gene ontology or GO analyses. And these essentially produce a really colorful table, as I'm showing you here. I definitely don't have time to go through each of these tables. And so I'm just going to summarize the function of these genes on the volcano plots itself. So for May, in response to heat, these upregulated genes were responsible for protein stability and cellular maintenance. And this makes sense because essentially, when our bodies experience high amounts of heat, parts of our cells and proteins can literally start to melt. And so these genes that are being upregulated here produce proteins that keep that from happening, that allow those proteins or those cellular components to remain stable and allow that copepod to survive despite being in this high heat stress. And then for these downregulated genes, they were mainly responsible for cellular division and growth. Essentially what this means is that these copepods are slowing down their growth rate in order to deal with this stress in a survive now and grow later response. Unfortunately, for August, long story, but my samples failed. So unfortunately, I don't have data to share with you from this month. But based on the fact that we saw very similar physiologic thermal limits throughout the upwelling season, we hypothesized that there's likely a similar genetic response in August in response to heat as there was in May. Now onto hypoxia, which, spoiler alert, I do have both months for, so luckily. In May, we saw a very muted response from our copepods in response to hypoxia overall, even though they were at their peak crate value, with only a small number of genes being upregulated or downregulated. The upregulated genes were responsible for cellular maintenance, which again, hypoxia can also stress proteins and genes, so proteins and cells. So these upregulated genes are also trying to maintain that stability in hypoxic stress. But interestingly, also growth, specifically the checkpoints of growth. That means that these copepods are essentially utilizing genes that are fine-tuning growth patterns to be able to continue growing despite being in this hypoxic stress. And then these downregulated genes were mainly responsible for maintaining metabolic rate. That means that these copepods are likely trying to lower their metabolic rate, lowering their need for oxygen, which makes sense as they're trying to grow and live in a more hypoxic environment. But overall, this small amount of genes means that they only need to make a small number of changes in order to live in these in order to live in this hypoxic stress. This is very different than what we saw in August, where thousands of genes were both upregulated and downregulated in response to hypoxia. But what's interesting is that they both upregulated and downregulated genes had very similar functions to the small number of genes that we saw differentially expressed in May. Essentially, what that means is that in both time points, the copepods are doing very similar things in order to respond to hypoxia, but because of this, well, I'm getting ahead of myself. But because they're likely spending greater periods of time in this hypoxic stress, they need to utilize a vastly larger number of cellular processes in order to deal with this hypoxic stress. So when we overlay these plots between May and August, we can kind of see here that copepods in August are likely more sensitive to stress because they're just using overall way more energy in order to respond to the stress than they did in May. And I'll talk about the conclusions of really what that means in general at the end of my talk. So for now, let's focus on project 3, which is my third and final project, which looks at how the environment throughout marshallized southern range impacts patterns of genetic variation and adaptive divergence between populations. So I've only been talking about marshallii on the Oregon coast, but in fact, they are distributed throughout the Northeast Pacific, and they play very important ecological roles across 3 major current systems, including the California and Alaska current systems, as well as the Southeast Bering Sea. Again, as plankton, they cannot swim against the pattern, against the direction that these currents are moving. And I got interested in whether or not this may drive separation between populations of plankton and result in what's known as population genetic structure between systems. So to explain this more, let's zoom in on the southern coast of British Columbia, where the Alaska and California currents diverge. You can imagine that if this divergence is strong, it might keep copepods, populations of copepods in either current system from intermixing with each other. And over time, just by totally natural random genetic variation, there could be genetic differences that drive divergence between these populations. We call this neutral genetic structure because these differences, as I mentioned, are totally random. They have no impact on survival or reproduction. They're just happening. They're happening independently of one another in each population. But as I mentioned earlier, oceanography is very complex in this region, and all it takes is one weird current or one weird pattern of wind to connect these 2 currents. And in this case, genetic structure is likely very low, or genetic divergence is likely very low within this population, essentially leading to one giant large metapopulation. And so with that in mind, I focused my first set of questions on neutral genetic divergence here. Again, that's genetic divergence that has nothing to do with reproduction or survival in these copepods. And I asked what patterns of neutral genetic divergence exist for marshallii and how do current systems separate or connect these populations. Alternatively, we can also have adaptive genetic divergence. And this refers to genetic differences that do have an impact on survival or reproduction in copepods. There is immense environmental heterogeneity throughout each of these current systems. And essentially, this could lead to populations becoming more adapted to local conditions in their own region compared to regions elsewhere. And so to explain this a little further, let's again zoom in on the southern coast of British Columbia, where there are 3 unique marine ecosystems that I want to share with y'all. So offshore of Vancouver Island represents the northernmost area of the California current, which means it does experience upwelling conditions throughout the summer, but interestingly, not as much hypoxia as areas further south. And then farther north into the Queen Charlotte Sound, this is part of the Alaska current system, where upwelling does not occur. And that leads to waters in general being slightly warmer and more oxygenated than in regions further south. And then we have the Strait of Georgia, which is a semi-enclosed area of the ocean connected to the rest of the ocean only through these relatively narrow straits. And what's more is that there are major river systems that deposit into the Strait of Georgia, making this region much less saline or salty than areas further south, or as well as warmer during the spring and summer than these other oceanographic regions. And so with these environmental variations, with this environmental variation in mind, in regards to adaptive genetic divergence, I asked, how does the environment potentially drive differences between marshallii populations, and which environmental variables could be important for this? So in order to answer these questions, we separated marshallii southern range across 6 unique oceanographic regions, from the Queen Charlotte Sound up here to offshore Vancouver Island, the Strait of Georgia, and then Washington, Oregon, and Northern California. So to compare how similar or different environmental characteristics are between these regions, I compiled over 20 years of CTD data collected at each of these stations that I'm showing you here in these dots in each region and during the spring and summer. And these variables collected in the CTD casts are temperature, salinity, and oxygen. And then I collapsed these data into a single environmental data set, which I analyzed using a method known as principal component analysis, which I'll get to. And then to create our genetic data set, we first collected copepods from throughout each of these regions. This was a very collaborative work between the Department of Fisheries and Oceans Canada on the coast of British Columbia and NOAA here on our west coast. It took about 2 years to collect hundreds of copepods, from which we took 200 for further genetic analyses. So with these 200 individuals, we extracted RNA from each individual, sequenced it, and then using these sequences, we created a database of genetic markers known as SNPs. A SNP is a single nucleotide polymorphism, and it essentially represents very small genetic differences between individuals that we can actually utilize to quantify levels of divergence. Overall, we found roughly 4,700 SNPs across these copepods from all of our oceanographic regions. But the tricky part here then comes to, how do we know which SNPs are potentially helpful for helping us answer our questions related to neutral genetic divergence, and which SNPs are important for answering our questions related to adaptive genetic divergence? For this, we can utilize an actual measure of divergence known as the FST, or fixation index. A value of 1, and for a value close to 1, represents a high amount of divergence in SNPs between populations, whereas a value closer to 0 represents a low amount of divergence in SNPs between populations. And we can utilize programs that can essentially model this variation in FST values, as I'm showing you here. And SNPs that are within this model, the model here is being shown by this blue line. When I say within this model, I mean under this blue line. So SNPs that are within this model are likely our neutral genetic markers, whereas the small number of SNPs that are above this line here on the far right represent outliers, which are potentially adaptive SNPs that we can use to answer our questions related to adaptive genetic variation. So let's get into the results here and focus on our environmental principal component analysis first. So principal component analysis essentially breaks down very complex data and is able to plot variation within that data. In regards to my data, we're looking at variation between oceanographic stations or oceanographic regions, and can plot that variation on a 2-dimensional plot. And I want to introduce this plot 1 dimension at a time. So here we'll start with the x-axis, and variation between regions here. So we have Northern California on the left, all the way to Strait of Georgia on the right. And variation here between these regions was largely driven by temperature and salinity regimes between them. So areas on the left are usually more cold and highly saline during the spring and summer, whereas areas on the right are warmer with low salinity. And this makes sense when you see that areas on the left are mainly those within the Northern California current, where upwelling is occurring, whereas way over there on the right is our Strait of Georgia, which I mentioned has a large amount of fresh water deposited into it from river systems and is generally much warmer than these other oceanographic regions. But now let's see what happens to these groupings when we add the y-axis to this, which is largely driven by environmental variation in oxygen levels. What we see here is that we have groupings based on whether or not regions were similar in temperature and salinity as well as oxygen. And as you can see, these areas further north sort of cluster together between the Queen Charlotte Sound and offshore Vancouver Island, as they are highly oxygenated compared to these other regions, but also are somewhere in the middle in terms of temperature and salinity. Interestingly, Northern California and Washington cluster closer together based on temperature and salinity regimes, but Oregon is way down here because it experiences significantly lower oxygen conditions than Northern California or Washington during the upwelling season. And then we have Strait of Georgia way over here, which forms its own, also its own unique cluster, given these really warm and low salinity conditions. So this is the clustering of our environmental regions. And now we'll look at clustering of our genetic data. So for this, we utilized a program known as Structure. And this is really cool in that it sort of assumes that we don't know how many specific populations of plankton there are between our regions. And so it can, so in order to sort of inform us of how many likely populations there may be, we can use a different number of hypothesized populations throughout this program. So essentially what this produces is a stacked bar plot where each column represents an individual. In the data I'm about to show you, you can imagine that we have 200 individuals or 200 columns along the stacked bar plot. And this is, again, just hypothetical plots, not my actual data. Each color here represents a population. And in this case, we're hypothesizing 2 populations throughout this range. So in the case where copepods are likely highly connected and therefore lack genetic structure, we likely see something like this, where each copepod has an equal proportion of each population within it across these different regions. Whereas if we had a case where Northern California current regions are separated from those farther north, we would see a unique population occur in each general region between the Northern California current and populations further north. And then we can simulate adding potential populations until we essentially see what seems like the most logical solution, or we get to the point that adding populations doesn't really add anything new in terms of our interpretation to how connected or not connected these populations are. So here's what we found using our genetic data set with our potentially neutral SNPs. As you can see, there are orange and blue from each population. So the presence of each population in each region. So overall, these patterns are represent a high amount of connectivity in a plankton between each of these regions. But what's interesting is that within Oregon and the Strait of Georgia, we have overall, there are a greater amount of individuals partitioned into this orange population than the blue, which is sort of unique compared to all of these other regions here. So that seems that populations in Oregon and the Strait of Georgia might somehow be more similar to one another than they are to populations elsewhere. When we add another potential or hypothetical population to this plot, it doesn't really add much in terms of our interpretations from 2 populations, and the same thing with 4. We see more colors here, again, purple with these 3 populations, and the 4th population is in green. But overall, they don't really make that big of a difference in terms of our interpretation than we did with 2 populations. And so it seems that in related to neutral genetic divergence, current systems may drive populations from Central Oregon and Strait of Georgia to be unique from one another, but overall, there seems to be about 2 ancestral populations here in our neutral genetic data set. But now let's move on to our outliers. Again, these are potentially adaptive. We don't know exactly if they're adaptive SNPs, but when we plot them with 2 populations, we see very similar results than we did to the neutral data set. But what's interesting is that when we add a 3rd population, all of a sudden these regions from the Northern California current become unique compared to those further north. So it means that areas in the Northern California current might lead to a more unique population than those further north. And also sort of what I want to point out here is that the population, this purple population is relatively sparse in Oregon than it is to Northern California or Washington, meaning that Northern California and Washington populations might be more genetically close to one another, or at least experience similar conditions than they do in Oregon. So there might be unique conditions in Oregon that are driving this region to be genetically different from those in Northern California or Washington. And then upon the addition of a 4th population, we see that the Strait of Georgia becomes, highly distinct from other regions throughout this area, from other regions that we are comparing. And so in order to save time, I'll just say that the addition of 5 and 6 populations didn't really add much here. And so essentially, when we overlay these 4 populations across our range, we see something really interesting when we compare it to patterns of the environmental variation. Essentially, these regions cluster extremely similarly as they do in the environmental data set, which has no genetics in it, as to our genetic data set, which has no environmental data to it. So Queen Charlotte Sound and offshore Vancouver Island cluster similarly together. The Strait of Georgia is its own unique cluster. And then what's super cool is that Oregon is unique despite being in between Washington and Northern California. So there could be unique selective pressures here, which we hypothesize is potentially this very low oxygen content in Oregon, and then these high temperatures and low salinities in the Strait of Georgia that are resulting in genetically unique populations throughout these regions, despite a high amount of connectivity. So these environmental conditions are driving natural selection, potential adaptation, despite a lot of gene flow between these regions. Cool. I basically just answered these questions a little early. So moving on to our conclusions. So I want to begin my conclusions with, again, sharing this question that I shared at the beginning, which is, how are plankton populations in the coastal Pacific Northwest being impacted by climate change? In Project 1, we learned that copepods can become more sensitive to hypoxic conditions over the course of a single summer. And in Project 2, we found that the biological processes that are impacted by this by this stress throughout the summer include mainly metabolism and growth. So the implications of this are that essentially marshallite populations may begin to decrease over time if they're putting less energy into reproduction and they're becoming more stressed out throughout the upcoming season every year, as hypoxia is expected to only get worse into the future with climate change. Similarly, in terms of these actual processes that they're changing, they are fine-tuning their growth, they're mitigating energetic processes. This could lead to their metabolic rates decreasing in order to adjust to these stressful conditions, meaning that they might consume less food and therefore carry less fat, which has two large implications. One, the phenology of the species, as I mentioned earlier, where they go into diapause over the winter. They could essentially be limited in the amount of time they're diapausing by having less fat to live on during this time of the year. But also, potentially more importantly, the fisheries that rely on this species as a high-value prey source may not be able to get as much fat as they were getting before when they were eating these copepods, making it harder for them to grow as well. And then finally, for Project 3, we looked at environmental variation. We found that environmental variation can play a significant role in shaping marshallite populations. And as such, that means that different populations may have unique responses to the same environmental stressors between regions. So if we sampled copepods in the Strait of Georgia, they may have responded very differently to the same stressors than our copepods off the coast of Oregon. And we need to invest more work into understanding how environmental stress tolerance varies between these different regions and populations of copepods. And so in conclusion, I found that zooplankton populations are being impacted by environmental change, but we need to know more. I focused on a single species, of which there are dozens throughout the coastal Pacific Northwest. And I showed how tools using ecophysiology and genomics can provide an important body of work that we simply don't have yet to inform management and to inform coastal fisheries about how zooplankton populations may be impacted in the future. And I'm very excited to continue this work as I move into my postdoc. Yeah. Okay, cool. And with that, I'd like to move into my acknowledgements. There's no way that I could thank all the amazing people who helped me through this PhD all at once, but I'd like to begin by first thanking the funders for my science, so the National Science Foundation Lewis and Maud Hill Award, through the Hatfield Marine Science Organization, the Integrative Biology and Research Award, and the Mary and Paul Roberts Fellowship. I would really like to thank my amazing graduate committee, beginning with my awesome advisor, Felipe. Felipe, thank you for bringing so much energy and enthusiasm into every one of my projects. Honestly, the very first time I met Felipe, I realized he was by far the most excited person in the room about doing science. And I just found it super inspirational. And frankly, since I started this degree, I've just been trying to be you every time I walk into the lab. So thank you. Kim Jacobson, thank you so much for taking the time to be a part of my committee. For those of you that don't know, Kim is not actually a faculty member at OSU. She works for NOAA out at Hatfield and has been a really incredible person for me to talk to in terms of thinking about what applied research really means and how the implications of my work can help support coastal management or folks or coastal communities. Kim Bernard, thank you so much for being my graduate council representative and for helping brainstorm ideas with me, research ideas, questions. Mark, thank you so much for being so accessible, for answering all of my questions, no matter how random. And Bruce, thank you so much for joining my committee. It was really special to have you on it, given how much work I did in your lab as an undergrad, and knowing that you could be here to help support me as I grow as a scientist. So thank you. I would also like to thank the incredible oceanographers who were totally the reason why I was able to get these copepods in the first place. Sam Zeman, Jennifer Fisher, Anna Baum, Josh Bowman, John Nelson, Moira Galbraith, Akash Shastri, and Julian Smith. Thank you all so much for helping me. These folks go out onto the ocean not only every month, but like literally every 2 weeks and are collecting these plankton. And it's thanks to them that we have this awesome knowledge of how environmental change impacts plankton populations overall and how I was able to get all of these samples. Special thanks to Sam and Jennifer. Y'all have been my mentors in oceanography since I first started this job, and I really could not have done this without you, so thank you. I'd also like to thank the research vessel crews that are keeping the boats going as we go out. Some of you may not know this, but there are much more than just scientists that go out on these vessels. So there's engineers, there's deckhands, there's crew members, there's stewards, there's cooks, there's janitors. It takes a large number of people to keep these boats going. And I just want to say thank you to all of the amazing people that I met on these cruises. There we go. That I met on these cruises. It was just one of the highlights of my PhD to go out and do this work offshore. And so thank you to all of you for helping support this work. I'd like to give a special thank you to Jeff Lawrence, who was the captain of the Alaka on my very last cruise, and also the captain of the boat that I went on my very, very first time as an undergrad on a boat. Yeah, it was really special. And I'd also like to thank one of my favorite crew members, Vlad the Wiener dog. He was very essential in keeping me calm in my data collection offshore. I'd like to thank the amazing Barretto lab members, both past and present. Thank you all so much for supporting me. Matt, thank you so much for helping me with going through all this physiology data. It was thanks to Matt that I was even able to get analyses going so quickly, and especially to Layla and Annika for being so welcoming to me, even though I basically was on Zoom this entire last year in our lab meetings. Thank you all so much for your support. Yeah, it's been such an honor, honestly, to be a part of the Barretto lab. I'd also like to thank my amazing undergraduate helper, Spencer Loring. He helped me essentially get off the ground with how to keep callus in the lab, how to keep them alive, how to keep them healthy, and start experiments on them. He's currently in Idaho right now as a freshwater biologist, kicking butt, and I'm so grateful that he was able to help me throughout my graduate research. I'd like to thank the amazing teaching teams that I was a part of with Carmen Harjo and Lori Kays leading. It is a significant amount of work that these teaching teams do to organize curriculum and, across hundreds of students. And it was amazing to be a part of that. I'd especially like to thank my recent class out at Hatfield that I taught by 450 from. This is a really cool course where essentially you get to immerse yourself in the Oregon coast for 10 weeks. And my students were fantastic. I had 30 of them, and they were so on top of it, so prepared, and made it able for me to teach them while also somehow being able to finish a PhD at the same time. I'd like to thank the integrative biology staff for all of their help in, of course, putting this together, but also making sure I was staying on track. Teresa Bolin, Tracy, Tara, and Melissa, thank you all so much for your support and helping me. Y'all are really the image of kindness and the heart of IB, and I just want to say thank you to all of you for all of your support. And I'd like to thank, And I'd like to thank this amazing department. As Felipe said, I've been here a long time, and I've made so many, I've made so many amazing friends and mentors to this department, and each of you had an important role to play in me being here. So thank you. I'm gonna take a seat. A special thanks to Cheyenne and Jesse for checking in with me and doing work Zoom sessions to make sure I was doing well and staying on track, as I've been glued to my desk for the last 6 months. And thank you to my wonderful community of friends back home in Half Moon Bay for keeping me active and reminding me to take breaks from time to time. This is my best friend, Cam. He's in the audience here today. Thank you so much for being here. Thank you for supporting me and just being my best man, my best friend. We've been friends since we were this tall, and been ambassador for a better best friend. Thank you, Cam. I'd like to thank, of course, my family. I'm so lucky to be part of a supportive and loving family. Thank you so much to my family members tuning in from all over the world today in the United Kingdom and Pennsylvania, Connecticut, California, and Tennessee. Your support has meant so much to me and helped me carry on through the last 5 years. Thank you so much to Lynn, my godmother, and her family for helping support me through all the hard times through my PhD. Me being here is thanks to y'all. Thank you. I want to thank my grandparents for being here, for always supporting me, for helping me to stay curious, and of course, reminding me that there's always something in every person that you don't know, and that everybody can teach you something. So thank you for helping me stay grounded and remember who I am, and for always supporting my curiosities. Thank you. Of course, I'd like to thank my family, my parents and younger brother, Kramer, my younger brother. Thank you for teaching me lessons in conflict management. They were very vital throughout my PhD. Dad, thank you for teaching me that nothing comes without hard work. Thank you so much for always being there for me. Mom, thank you for always believing in me. I know when you dropped me off here for my freshman year 10 years ago, you told me I would leave my mark here, and here we are. And of course, last but never least, I'd like to thank my amazing wife, Caroline. Thank you so much for always supporting me through every moment of this PhD, for keeping our four-legged family going. There were so many times where I wasn't sure if this was going to happen, but you always saw the person standing here. So thank you. I love you. And thank you again all for being here today. As you can tell, I've been thinking about this a lot over the last 5 years, and it's just so important to me. The first place I ever saw the coast was Newport, actually, when I was a little kid. And it means so much to be able to have been able to give back to a community that's given so much to me. So thank you all for being a part of that here today. And with that, I'll take any questions.
Still, we can get kicked out. We have a few minutes. And we have to go to the tomorrow.
Yeah, then the fun starts.
Well, really cool results. It's really impressive that you can collect Calanus marshmallow from the environment and then take it to the lab, still haven't survived and do these experiments. It seems like it could be complicated by the fact that, from what I understand, you have years where Calanus pacificus is dominant, years where Calanus marshmallow is dominant, probably based on temperature and upwelling.
Right.
And I was wondering, did you have to sample multiple years to get specimens for your experiments? If you did you consider interannual variation in the responses to temperature and hypoxia?
Yeah, it's a good question. So for folks over Zoom, what Luke is asking about is the other species of Calanus within this system, which I didn't mention, Calanus pacificus, which looks almost identical to my copepod species and makes it very difficult to speciate them. And in general, the patterns that Luke is asking about is in warmer summers, we typically see more Calanus pacificus over the continental shelf during upwelling season than we do marshalli. And to answer your question, so we developed a genetic method to be able to identify them surefire between each species. And essentially, I didn't know at the time if I was going to be collecting Calanus pacificus or Calanus marshalli. All I knew is I had Calanus. And so after data collection, I basically just sampled as many individuals as I could. And then after data collection, speciated them based on this genetic technique. And to be frank with you, I got lucky that there was an abundance of marshalli throughout the single summer that I sampled. If it had been a relatively warmer summer, maybe this project would have been about Calanus pacificus. So yeah, it was luck, but also through the support and sort of guidance from many of the oceanographers that I mentioned, including those that I didn't, about how to keep them in the lab, when are marshalli most likely active versus pacificus, and where to find them. So it was through using this network that I was able to sort of pinpoint where marshalli were most likely to be. Yeah. Yeah, Shane.
I guess a follow-up question to that is, you mentioned that you looked at marshalli specifically, but how representative of species is that for like?
Yeah, so they are a dominant species in this sort of cold. So as I mentioned, marshalli are sort of the cold water species, and pacificus are the warm water species. And so They are the dominant copepod species in this cold water. As our climate changes, it's kind of tricky to know if Pacificus might become more dominant in the future as our waters warm. Potentially, if I had to hazard a guess, I would say maybe not, because when upwelling occurs, it's these cold water masses that are coming down onto our coast from these northern areas, and these are rich with marshali. When we've seen Pacificus more on our coast during the summer, it's been in unique situations where upwelling isn't as strong, and so these populations are not moving down. But if upwelling is supposed to get stronger, then we'll likely continue to see marshali, but whether or not they can adapt to this warmer cold water in the future, if that makes sense, or this low oxygen water, is something we don't really know. So they are the dominant, but then over winter, as I mentioned, marshali dive deep, and that's when we find Pacificus on our coastal shelf when upwelling is really not occurring. Yeah. Francis, did you have a question?
Yeah, Kris, hey, congrats, great work. I'm curious about the seasonal variant P crit, and not a lot of people actually look at P crit over time.
Yeah.
It's usually one and done.
Right.
I think you're interpreting that there's some activation that's going on, that this is within population process. But your third chapter suggests that there's an admixture of different sources of different genetic stocks.
Yeah.
Are you able to differentiate whether the seasonal dependence is something physiological happening at the scale of the individual?
Yeah.
Or is it just gene flow?
That's it. Yeah. So as I mentioned to Cheyenne, we have populations of marshali moving south throughout the upwelling season. So we could have been sampling different populations, even though we were sampling the same region. It's difficult for me to know that without more genetic markers to be able to say, oh, this copepod came from the Strait of Georgia or Western offshore Vancouver Island. So it could have been that, yeah, it wasn't necessarily the same population that was becoming more sensitive, but instead, these populations that are moving into Oregon throughout the summer are actually the ones that are more sensitive. And I don't have the resolution in my data to tell that, to know whether or not we really were sampling different populations. But yeah, like I said, different populations could be responding to the same stressors differently. Regardless, though, what we saw in August are these sort of dominant marshali populations on the Oregon coast. And so whether or not they're from the same region or from different regions, they are more stressed out in hypoxic conditions at the end of the upwelling season. And so I think the implications in terms of the ecology of the system are still valid.
One more.
Yeah, Sean?
So, this is awesome. How long do they live?
Yeah, it's a
And
very
then-
good question. Yeah.
See, some of your tests were based on their mortality.
Yeah.
Can you directly observe changes in their longevity in the environment?
Good question. So it's incredibly difficult to track a single individual growing up in the environment. Essentially, that model I showed you where we see young individuals, in the surface, and then, older individuals deeper that are brought back on the shore. That has been through intensive sampling where we sample different areas of the surface. We sample different areas at depth. And we essentially look at the abundance of each life stage throughout the upwelling season. And we do this pretty much I think at that time, like literally every day throughout the summer. And so those are just sort of the general characteristics of life histories. So we think that there may be anywhere from 3 to 6 generations per upwelling season on the Oregon coast, but they're highly dependent on temperature. The warmer it is, the faster they grow. So populations up in the Bering Sea, we think we only get 1 to 2 generations per summer because it's so much colder up there. Regarding the lab situation, I did experiment with trying to raise them from hatch all the way to adulthood, but I don't think I had quite the right setup. Essentially, I was just keeping them in these little jars, and they need a big wide ocean that is just mixing around and going, doing all sorts of things. And so in the lab, folks who have gotten them to grow, they usually live anywhere from the generation time is between a couple weeks to a month and a half. But the only sort of outlier is that if you get to a stage where upwelling conditions are about to end and they go into diapause, they can live for months at that life stage. So, it really just depends on whether or not, it's not obligatory. They don't have to go into diapause. That's why we have multiple generations throughout the summer. But if you are a copepod that's about to go into diapause, I'm really interested in looking at how does that impact your stress tolerance over time. So they can be anywhere from a few weeks to months, depending on whether or not So yeah, a long answer.
All right, y'all. Thanks so much. Grab some pastries on the way out.
Yeah, please, please.
Yeah, well, your dog. Yeah. Yeah, she's going today, she's. Yeah.